Thanks, Romana. Thanks, everyone, for coming. So those who know me or see my affiliation, you guys may have some questions. One, like, why am I here? Um, I asked that to myself as well. I live in San Diego, but my connection to the Great Lakes Bioinformatics Conference started when I spent some time at Ramana's lab back in 2012, I think 2012. So that's where I started coming to the conference, like organizing some sessions, meeting good friends, and then I still try to attend when I can, although it's a bit of a trouble. The other question, if you know or heard about what I am doing or what our lab is doing generally, which is the 3D genome organization, and then why am I in like isoform, level analysis type of session? So again, one answer is because I spent some time with Ramana, and then the other answer is the data has taken us towards that path, right? So today I'm going to talk about that story. That we have been working for more than 5 years by now in the lab, and that started as a completely different direction at a different direction, but have been taking us towards this. So I will give you a little bit background in what we do. This also becomes relevant. So my lab is really interested in understanding the 3D organization of the human and eukaryotic genome in general. So we in each human cell, we have about like 6 feet of DNA, and it's packed tightly and in an organized way, and then that DNA folding and packing is not happening in random, and then it changes from cell type to cell type. So it does have functional influence on how the genes are going to be, cells are going to function. So there are different layers to this organization, and then the coarse level layer is that like chromosomes generally tend to be folding within themselves and closer to regions within each chromosome than the other. That's what you're seeing in the first big heat map, that each diagonal square is a chromosome. When you zoom in, take a chromosome and zoom in a little bit more, maybe at the 100 kb bin resolution, you're still looking at a couple megabases here, then you start seeing these two broad coarse definitions of chromatin. Active chromatin, which with high C we call it A, that's arbitrary, and inactive chromatin, which we call B. So that's chromatin compartments. So if you zoom in, take a couple of those compartments or one of them and zoom in a little bit more and start looking at the higher resolution, then you start seeing again these diagonal square blocks, and then some of them you can see that some of them are fuzzier with no dot at the corner, and then some of them have a dot at the corner, right? So these domains are called topological domains, and these are regions of DNA that interact highly within, but not with the across the two such separate domains, even though they are neighboring. And then at their boundaries, and sometimes in the inside, if you look at the little wiggly figure there are what we call chromatin loops, right? So when they're at the boundaries, they're generally associated with structural proteins, and then they demarcate this insulation of this domain. And then sometimes they are within these domains, and those generally correspond to specific interactions between regulatory elements, some of them distal, and then the potential genes that they regulate. So my lab has been working on this ever since I was in Bill Noble's lab, and then we have developed different tools both for the analysis of compartments, identifying and assigning function to these chromatin loops also, and one part today that I'm going to talk about is about genomic rearrangements, because while analyzing these type of contact maps, what we realized is that these proximity patterns are heavily influenced by one-dimensional distance between two regions, right? If two regions are two loci are right next to one another, they by definition are going to have a lot of interactions in this type of experiment. So that's the heavy diagonal that you are always seeing, right? And then that becomes really kind of like a something that you can opportunistically use when the cancer genomes have rearrangement, because now two regions that are supposed to be very far away will be juxtaposed because they were broken and then like put together. So we and others have developed algorithms that use those type of patterns from high C maps to identify uh, these structural variants, including translocations, insertions, deletions, and then we stumbled upon a little bit more of a difficult case where this is an artificial system where you have mouse embryonic stem cells, and then you're trying to have a model for Down syndrome, which means trisomy of chromosome 21 in human. In mouse, it's chromosome 19. So mouse says it's two chromosome 19s, and then now like you're trying to insert a human chromosome 21 into mouse ES cells. And then you do that by first irradiating human chromosome 21, which kind of like makes it shatter. And then after this, once you electroporate into the mouse ES cells, then it kind of gets put back together, the pieces, but you can think of it almost in a hurry, that it doesn't get put together in the correct order. So what it then gives you is a high C contact map like this, where if you remember the main pattern, the diagonal would be what you mainly are seeing if this was a normal chromosome, not shattered. Here you have a whole bunch of off-diagonal patterns. So we found this high C data when we were trying to work on these genomic rearrangements, and then Abhijit Chakraborty in my lab then developed an algorithm that can take a contact map like this, identify the boundaries of such potential breakpoints, and then essentially shuffle it back together, right? So this is important because then now you really get the DNA sequence that is specific to this cell type, or in cancer genome, when these type of events happen, that would be specific to that patient, because each patient's shattered chromosome gets put together in a different way. So what this is, that was just a model, and then what this is relevant to in cancer is a concept called chromotripsis. And this concept was a little bit kind of like not on the radar for quite some time, but back like maybe 10 years ago by now, or 6 years ago probably, there was a paper that came and then done whole genome sequencing on about 3,000 different cancer genomes, and then said that, oh, that like weird rearrangement called chromotripsis that we were estimating is like 1 to 2%. Well, that's actually 20 to 30% of the cancer cell, right? So all of a sudden, then there was a lot of interest, and the ecDNA people followed up, and so on and so forth. But this is a catastrophic event. It's just like this: the chromosome gets shattered, and then puts back in an order that is really difficult to understand. And then like what kind of events happened is important, because genes may be fusing to one another, genes may be breaking, non-coding elements may be coming together, like enhancers that are not supposed to regulate an oncogene may be coming together with oncogene. So then like after solving that toy problem, then we are like, okay, what are the cancers that that have chromotripsis events that are happening? So interestingly, the path took us to a pediatric cancer, which I will talk about today. But in general, pediatric cancers are actually quite different than adult cancers. And then one major difference is that their mutational burden is quite low compared to adult cancers, right? There are a lot of pediatric cancers which have, less than 1 in 1 million base pair kind of like somatic mutations. But the one that we are going to study today is not, and the one we are going to study is a leukemia of B cells. So it's an acute lymphoblastic leukemia, and then you can see that like it is in the hematopoietic lineage differentiation. This happens at the lymphoid stage, and then the one that we are specifically working in happens at the pre-B cell stage. Right, cells are committed towards being B cells, but they really haven't gone all the way. They are early in that, and then these cells acquire these mutations, and sometimes this chromotripsis. and then become tumor. So this is a subtype called IM21. It's an amplification of chromosome 21, and it leads to this type of huge blowing up of the chromosome 21 with lots of amplifications. Its a characteristic pattern is this stepwise pattern that like different kind of levels of copy number alterations happen, and then there's a region that is commonly amplified that is shown there. And then these people have shown with microscopy, and then when you do, the type of assays that we like, which is high C, you really see it also as this shuffled pattern in the chromosome. So this is a high-risk subset. This is luckily study from 15 years ago, but as you can see, it's a horrible prognosis for these kids. But luckily right now, they are identified early on and then being treated with the high-risk protocol. And then when treated with the high-risk protocol, they still have a higher risk of relapse, but their survival rate is coming close to standard risk protocol on the low-risk subtypes. So you really need to know it to treat it with with that kind of protocol. It's a rare subtype, so it's not as well characterized. It's hard to study. It's rare, and then it's aggressive. So how chromathripsis happens is a very interesting concept. I don't have time to go over it, but if you look up something called breakage fusion bridge, you'll get some sense of it, that it has something to do about telomeres fusing, and then the rest of the cell cycle going crazy after that, leading to this type of shattering. So my lab is mainly computational, but we also have a wet lab where we can do some, at least genomic characterization with help from the cores and others at LGI. So what we have done here is that we put together a cohort. So first of all, we got some samples, 12 IM21 samples, and 8 non-IM21 B-cell leukemia samples from Children's Oncology Group to do our own experiments. And then we also collected recently published data to really boost up the power of our cohort. For the whole genome sequencing, we have 120 or so different patients. For RNA-seq, we have 40 or so. And then we've done our own single-cell RNA-seq, high-C, and ATAC-seq on a subset of these samples. So the first thing we looked at is whether we can see some heterogeneity or some different subtypes within this subtype, essentially. So for that, Abhijit first looked at the copy number variation, just using the whole genome sequencing data that we generated and compiled across these 120 or so donors. And then when we looked at it, when we clustered simply the copy number, profiles, you can see that there's 3 different profiles. There's one that has a deletion on the Q arm of the chromosome 21, that's cluster 1. There's one that's amplification on the Q arm, and then there's one that's a little bit in between. So we thought these 3 different types may correspond to something functional or some functional difference. When we looked at the gene expression, though, of these 3 different subtypes and compared them to one another, we really didn't find much of interest or anything that's unexpected. So you certainly find, like, where that deletion is, there's a whole bunch of genes there, and those are by definition not expressed anymore. So you find them, but really not much to see here. So then we concluded that maybe looking at the copy number based classification may not be the right thing to do. So how about look at the gene expression itself? And then we first combined our cohort with a published cohort from Charles Mulligan's lab, and then the red dots are IM here. So you can see there's some heterogeneity, and sometimes they have secondary alterations too. And then something that we are trying to understand is whether chromathripsis comes first and other things like that ETV ranks 1 translocation comes second, or vice versa. But one thing we did is that we looked at a previous cohort, that's 2023 publication in Blood, and then the cohort that we have put together, and then we've done differential expression analysis of comparing this subtype to all other leukemia, and then came up with about 1,000 genes that seem to be going in the same direction, up or down, across these 2 different cohorts. So trying to come up with a consensus list so we can do something with it or prioritize it further. In addition to the bulk RNA data, we also generated the single-cell RNA-seq data, and then we combined this with some published, healthy bone marrow B-cell progenitor data as a control. So here you have the colors are a little bit difficult to tell. I wish we picked a different color scheme, but you have some IAMP21 samples, 10 of them, and then some controls together with healthy bone marrow control. So when we did differential analysis here in the single cell domain and compared to those 1,000 genes, we further narrowed them down to like 277 or so that are, again, like dysregulated specifically in the IM. So when you then go look at what these differential genes are, and then select the subset of them with high expression, you can see that on the right is IAM21, and then you can see that there's a very good pattern of like a distinct expression for them. And some of these genes are important. There's interferon gamma receptor, there's a kinase, there's SON, which we'll talk about. So there's a lot of transcription factors and important genes that are distinctly expressed in this subtype. And then that was genome-wide. If you go look at the chromosome 21, where there's amplification, you also see a similar pattern. This is a little bit less unexpected, I guess, but then you see overexpression of a number of genes that are associated with this amplification of chromosome 21. You then ask the question, what are these genes? What are the genes that are upregulated in this aggressive subtype? And then something that came up again and again was NF-kappa B signaling. So if you don't know much about NF-kappa B pathway, it's one of the most central pathways to inflammation, cancer, autoimmunity. On the one hand, it coordinates response to viral infections. On the other hand, it does give resistance to apoptosis, which some cancer cells use it to their favor. So if you look at some of the NF-kappa B pathway genes, you see them specifically expressed in the IAMP patients and not in the patients that have a different kind of leukemia. And when we looked at only the IAMP samples and see like what distinguishes or what explains the heterogeneity within this subset, you see these NF-kappa B genes and so on and so forth come up as on the first principal component, come up as genes that explain most intra-subtype variability. So then we started going down this path of like, okay, what's happening with the NF-kappa B pathway? There's a canonical and non-canonical pathway. We put together all the genes. We looked at the expression in a single cell. So all the results were bulk so far. And then now we look at the single cell. Then you also see NF-kappa B high versus low, or we call it dependent, independent kind of pattern for each patient here, which you can see much better with the kind of like a UMAP or PCA type of analysis, dimensional heteroduction. So when you look at the patients, only the IM patients, and then when you look at where these NF-kappa B high patients are versus low, they do naturally separate. There are a couple patients which are interesting cases that some of their cells are NF-kappa B high and some of them are NF-kappa B low, which is it has something to do with the clonal heterogeneity, which I'm going to talk about a little bit. So when we did the differential analysis one more time in the single cell domain, we got, as expected, we got the NF-kappa B, but we got the mRNA splicing and spliceosome. So I'm slowly going through this like isoform, if you see where we are going, right? So that then this was kind of like unexpected, but knowing that some of the core components of nuclear speckle are upregulated, so it is also not completely surprising for us. So then we looked at these mRNA splicing related genes within the IAM cohort, but separating the ones that are NF-kappa B high versus low. And then as you can see, these genes the splicing related ones are highly upregulated in the NF-kappa B high subtype. So some of them include, those genes include the SRRM genes and then the SON. So these genes are core components of nuclear speckles. SON and SRRM1 are the two key components of what makes up a nuclear speckle. And nuclear speckles are where the RNA gets spliced and processed. So people have shown, and we have a collaboration ongoing where we are seeing that the SON downregulation, or the nuclear speckle component downregulation and disruption, has huge impacts on the splicing landscape. Here also we are seeing that it's the other way around. Overexpression of these proteins have also quite a lot of impact on the splicing landscape. And then when we did the splicing analyses of the differential splicing analysis of the NF-kappa B high versus low populations, and then intersected them further with the differentially expressed genes, which, after listening to Romana's talk, which maybe we should do it also for the genes that are not differentially expressed but differentially spliced, but there's a lot of them in this case. So when you look at them again, the NF-kappa B genes came, and NF-kappa B pathway genes came with the differences in their percent spliced in the exon inclusion in their transcripts. So there's certainly something going on that impacts nuclear speckles, and nuclear speckles impact genes that are splicing of genes that are related to the NF-kappa B pathway. So my lab, one of the new toys we have, something that we are excited about, is Oxford Nanopore sequencer. And then what we have done, expensive toy, by the way, though I'm not suggesting you buy it. The service contract is very high. So one thing that we were able to do with that is that we took pre-B cells. Unfortunately, IM21 doesn't have a cell line model. We talked to the person who discovered IM21, Christine Harrison, and then she said her lab spent 10 years or so and then gave up. For some reason, you cannot create a cell line, or so far it hasn't been able to create. We took another pre-B cell line, at least matched in terms of hematopoietic stage, and then overexpressed using an external construct overexpressed this gene called SON. And then we did long-read sequencing with or without this overexpression. And then what we then saw was that we saw also both differential transcription usage and differential expression. And then when we looked at the genes, those 22 or so here, which have differences in splicing and were upregulated in the NF-kappa B dependent case, and I looked at them, some of them, here it's a bit of the raw data. We have done 2 replicates, and then you can see that some of them, you have an upregulation of certain transcripts, certain isoforms in the overexpression, and some of them you see a downregulation. And then when you look at what are these 22 genes and what they are enriched in, we did then confirm that these are genes that are associated with nuclear speckles and then the mRNA splicing and so on and so forth. So our working model on this side is that the aberrations on the chromosome 21 lead to overexpression of genes, including the SON gene and others that are associated with nuclear speckles, and then this dysregulates the mRNA splicing, and including genes that are related to NF-kappa B pathway. And I'm not talking about it here, but what we are also seeing is some upregulation of genes that are not supposed to be expressed in B cells, like NK cell genes and T cell genes that are never supposed to be expressed in T cells, in B cells, which is what we are studying, start popping up as expressed in these. So likely this constitutive NF-kappa B activation leads to turning on some enhancers and some promoters and genes that are that are extra lineage, and then this leads to 2 subtypes. This is again a very simplified analysis. So taking things back to what we really like is chromatin looping. So we have done Hi-C in these 10 donors or so. We analyzed Hi-C data for more than a decade now, but first time we have generated ourselves. And then when we looked at the looping, and then looping differences between the two subsets. What we have seen again, this is completely now it's a little bit you already pre-subset, but then the genes, the looping changes, then you see again the NF-kappa B genes coming up. So it may not be clear here, but you do see specific loops at the NF-kappa B related genes that are only present in the NF-kappa B high subset and not the other one. So we looked at the genomic rearrangements and then how they look in both 3D and from whole genome sequencing by looking at the ones that are in the intersection of these two methods. First of all, we identified not all of the patients have chromotripsis, but most of them do have. So you see the ones with lots of off-diagonal patterns, those are chromotriptic. We have identified an unexpected chromotripsis event on chromosome 5. This is outside chromosome 21, and I don't think anyone has reported it before. And then we have identified a couple different interesting deletions and translocation events that involve chromosome 12. And then when there is a translocation, this is what you see. You see this, when there's an interchromosomal translocation, you see this butterfly pattern, which is distinctive and easy to identify. So what do these rearrangements lead to? So one thing that, this is one example, again, there's a couple more. One thing that that's fascinating is that sometimes certain genes that are not supposed to be expressed at all come together with a B cell related gene or a B cell related transcription factor. PAX5 is one. And then if you look at the uncorrected contact map on the original, you see this translocation. When you correct it, you see that, like now they have come together. And then when you look at this patient's gene expression, you see that this mapped gene that's not supposed to be expressed is expressed at very high levels, almost at the PAX5 level, because now they have fused with one another. And in the rest of the samples, whether IAMP or not, this gene is not expressed at all. And that was the bulk RNA-seq. This is the single-cell RNA-seq. You see that sliver of mapped expression, that is exactly the cells from that specific donor. There are cases where you also have like these fusions create what we call neodomains, neotads. So you can see that like between chromosome X and 12, that translocation leads to a creation of a new topological domain. And then when we looked at the single-cell RNA-seq data and done copy number calling from RNA-seq, we don't have single-cell DNA-seq, but RNA-seq data you can use at a coarse grain to call copy numbers. When we looked at it, we were able to see some patients have, clonal heterogeneity. So they have subclones, one with that kind of translocation and one without. So again, going back to our, the simplified model, we have, I think, tied everything together, going from just taking one cancer subset all the way to, transcriptome, all the way to genomic changes leading to transcriptome, leading to splicing changes, and then also changes in the epigenome and 3D genome organization. So I'm going to skip that slide. I already summarized everything. I think I will thank people who did the work. Abhijit has been working on this for more than 5 years. He's the driving force. Laura has been helping with the analyses. Nirmalya and Adam have generated some of the data. We got some initial funding from this, from a small Spark Award, some from Moores Cancer Center and Conrad Previs Foundation in San Diego, and samples came from multiple different places. We thank our collaborators at ALGI and Mayo Clinic, and I will take any questions. Thank you.
That's great, Ferhat. So, okay, is it when the looping happens, is it possible to infer at the alternative promoter level the interactions?
Yeah, good question. Yeah, I mean, this data isn't as deep, but the newer methods, either like Hi-C coupled with ChIP-seq or Hi-seq coupled with capture or micro-C type of methods, they will be able to distinguish alternative promoters from one another, especially if they are more than like 5-10 kb away. Certainly. At the very 10 years ago, after leaving your lab and starting my lab, I looked at these and then the resolution wasn't there. But right now it is. It is certainly. And I think there have been people who
So my other question is, the NF-kappa B pathway, you looked at the splicing, but did you look at the also, maybe they may be affecting the alternative promoters again?
We haven't looked at that. That's a good
particularly
suggestion. Yeah, we can go look at that.
the pathway, that thing.
Yeah,
That's,
yeah.
I guess.
We will look at that.
Okay.
Thank you.
Anyone else?
So really interesting work.
Thanks.
Have you considered looking at ATAC-seq data from this and looking at maybe base pair level footprinting with Chrome BP net? And if you do that kind of analysis, do the footprints reflect NF-kappa B or AP1 transcription factors? And
that's something that we really want to do. But one thing I didn't mention, but we have utilized all of our samples, and our single cell, our ATAC-seq data was single cell and only on couple patients. So it is really not deep enough for us to be able to do that. But that's exactly what we want to look at. We are talking to other clinical collaborators who are going to be able to hopefully give us some more samples, and exactly what I want to do, like see whether these extra lineage genes, whether they have NF-kappa B related activation footprints. It's tough to work with clinical samples. In my case, going from bioinformatics all the way, jumping into clinical samples all at once, there were some learning curve over there. So lessons learned. Thank you.
Really
That's
cool.
great.
Thank you.
Thank you.